Hello everyone. So today I'm going to be guiding you through the process of analysing multi-tiered or multiple images of gels and blots all at the same time within our Fretix 1D software. Hello everyone. So today I'm going to be guiding you through how you can analyse either multi-tiered gels or, if you've got a large format scanner, multiple gels at once and compare them to each other within our Fretix 1D software. So if you've seen my other videos, you'll know that this is the image setup window that will highlight to us any areas of overexposure within our gel or our blot within this section here, a 3D view of our bands here, and we can change the color, change the contrast and the display options within this window here using the display options here. So I'm going to do this quite quickly because I'm going to presume that you've seen my other videos that kind of go through automatic lane detection and manual lane detection and things like that. But I'm going to focus specifically on how we define multiple different lane boxes within our software and how you can use the automatic lane detection in different areas with kind of different parameters and things like that. So if we come down to our lane section of our workflow, it's where we define all of our lanes within our gel. Now this is quite clearly, I think, two separate gels, but this would apply to a multi-tiered gel as well if you are using those. Now, if I just click on detect lanes here, using the automatic detection algorithm, as you can see, it's not done the best job. It's been a bit confused by the gap between the lanes, and if I had a multi-tiered gel, so this was all one continuous gel, what is likely to happen is that it would just detect all of these lanes as very long lanes rather than separate them off. Now what we can do is, if we click on use AOI, we can actually give our automatic lane detection algorithm points to look in for the automatic lane detection. So if, say, this was, I'll do this tier first. We can draw the area of interest for our automatic detection algorithm, bring this down below the sample wells. And I'm going to do this roughly because it would be very boring to watch me fine-tune this for you watchers, but this is just an example to show you what you can do with our software. So now if I click detect lanes, you can see that the software has done a much better job at being able to pick out those individual lanes. If I wanted to then go and introduce another area of interest and detect within those, what I can do is I can drag my initial area of interest, and it works very well for this image because they are the same kind of gel in two different tiers. I can drop that down and run the detection again, and I've now got two separate lane boxes that you can see on the right hand side here. We've detected 25 lanes, and then from here what we can do is go on to edit those lanes if they're not a perfect match, which these ones aren't, or if I was wanting to define this manually, so say I have this two-tier gel here, or these two gels next to each other, but actually I want to separate them into four. So I've got samples here, different samples here, different samples here, and different samples here, and I want to quantify them, compare them, normalize them to each other, and treat them essentially as separate experiments. So if there were four separate gels, and I've just used my large format imager to image them all at the same time using the same capture settings. What I can do is, that was 25, which doesn't divide by 2 very well. Let's say this was two 12s. I can draw my lane box from here, release my left click, that's one lane box, start another one, have that here, you can see it's labelled box 2 already. Draw my third one here, and I think you can see where this is going, and then draw my fourth one here. So we can have all of those separated out and analysed as if they were individual gels or a multi-tiered gel, rather than just being able to consider one image is one gel or one blot. So that's a really quick run through. The rest of the analysis would be very much the same. You can see my other videos. We can do our background removal techniques. We can do our molecular weight assignment, and you can have a separate molecular weight for every lane box that we've implemented, as you can see on the right hand side here. So if I did have 4 different gels and they had different molecular weight markers, things like that within my scan. So say it come to the end of the week and I wanted to scan everything all at once and then separate it using the software, I can then kind of grab the molecular weight marker, Then on box 2, I can grab a different one and apply that. And then on box 2, etc., etc., I can keep going through and adding separate molecular weight markers for each box and treat them as separate gels, even though I've imaged them all at the same time. Come through to the bands mode, run my detection, do my detection, all those kind of things, come through to the results and export it just as normal. So that's a really brief run through, and I just wanted to highlight the fact that our software does support people that are doing large format multi-tiered gels, or if you wanted to scan all of your gels at the same time at the end of the week, for example, software will allow you to segregate those into separate experiments. If that's the application that you're currently performing, the software allows you to use that option, so it gives you that option within your experimental arsenal. As ever, thanks for watching, and if you'd be interested in trying out a free trial of Fretix 1D using your own images from your own lab, please check out the links in the description below.